And we record now. So welcome again, everyone, to our European Society for Marine Biotechnology webinar series for 2020-2021. I'm going to share some slides just to introduce you very briefly to what the ESMB Society is. So if you bear with me, I have a few things on at the moment. All right. So this is our logo. This is the ESMB logo. And the society is there to promote the exchange of scientific information, training, for example, in MSc students, and cooperation of its members to the benefit of society and the environment. Now, the society was actually funded in 26th of April 1995, so a few years now is running, and it was created because there was an urgent need to actually bring together marine biotechnologists across across Europe, and this was actually funded in France. Obviously, now it's pan-European, and actually we do have people joining at times across the world as well. Our activities are, for example, we do organize conferences and webinars as the one you are a part of today. We have meetings and trainings between undergrad, actually MSc students, for example, and PhDs, and we use the Erasmus Plus system quite frequently. We have access to the Journal of Marine Biotechnology, of course, to the members of the ESMB Society, and you will be receiving newsletters as well. The webinars and the conferences are free to join to everyone like today. So this is our website, and this is, you can have all this information, and actually this recording will be uploaded on our webinar tab at the end. So another thing I would like to say today, we're going to have two speakers, which will be introduced by Dr. Concetta Messina, who is our vice president. For the time being, I would like you to have your microphones muted throughout the webinar. And we will have the two speakers, and at the end, we can take questions. You're more than happy to write your questions on the chat room, and then we can read them out, but you can always actually speak out at the end using your microphone. Any of that will be fine. Hopefully, there'll be 20 minutes for each speaker, and then around 10 minutes at the end for questions and answers. And obviously, I will keep it for a little bit longer on. I have like a few minutes if we have more questions we need to add. So with that, I'm going to stop sharing, and I will ask Dr. Messina to introduce our guest speakers.
Good morning to everybody. Thank you very much, Maria. Welcome to everybody on behalf of the President, Professor Sergei Dobrezov, and on behalf of all board of the European Society for Marine Biotechnologies, and that had the idea to organize these cycles of webinars within this year, by inviting guest speakers from around the world in order to present their work that is in line with the field of marine biotechnologies. Topic, as you have seen, range from many aspects, within many aspects of the marine biotechnological potential. And the session of today was realized thanks to the sponsorship that the European Society for Marine Biotechnologies gave to the international conference on marine lipid that is organized for next July. Originally, the conference was organized for the past summer, but due to the COVID emergency, it was postponed. But we had online version in the past November, reduced version of the full conference, and within this conference in November, a young researcher presented their work, and European Society for Marine Biotechnology gave the sponsorship to this conference and decided to grant for prize the best oral presentation within the topic of the lipid applied to marine biotechnologies. Within this presentation, we selected two work presented by two researchers that were Ena Pritisanac, that we invited to present also her work today, and Anna Bispo, and together with her supervisor, Professor Narcisa Bandarra, that is also today with us presenting their work. So we are very pleased to have today these speakers, and I'm going to briefly introduce the two speakers. Professor Narcisa Bandarra is a researcher, and she holds habilitation in ocean science by Faculty of Science of Lisbon, and a PhD in biotechnology at the Instituto Superior Técnico of Lisbon. Actually, she's the head of aquaculture, upgrading and bioprospecting division from Portuguese Institute for the Sea and Atmosphere. And she has a lot of experience, scientific work in the area of upgrading of fishery and aquaculture product, with special focus on biochemical nutritional value of fishery and aquaculture product. And of course, so the result of her scientific activity together with her team is reflected in the publication that more than 170 scientific articles in international journal. She participates in 55 projects, national and international, oriented for research and development, and she supervises undergraduate, master, PhD student from national and international level, and she is the Portuguese focal point of the European Fish Technologists Association, and she was invited as expert to the high-level panel in ocean economy. And I kindly ask Maria if now we can continue with the presentation of Ena, or it's better that I introduce later. What do you think?
If you continue with introducing the second speaker,
okay.
and then we continue with the talk, that would be great. Thank you, Consent.
Okay, thank you. The second speaker is Ena Pritisanac, that is based in Croatia, and she graduated in the Department of Biology at the University in Osijek, and got the title of Magister Biologist. She worked as a volunteer in the Laboratory for Water Ecology and worked on popularization of science, the zoo and aquarium, where she led and participated in many workshops in the same city. And following the enrollment in the graduate school of biology, she became a leader of the student project Spreading of Invasive Species Aedes japonicus in East Croatia, for which she won the funding. Currently, Ena is working as an assistant in the laboratory of marine ecotoxicology at the Center for Marine Research in Rovini, and she is in the second year of interdisciplinary doctoral study of oceanology at the Faculty of Science in Zagreb, in Croatia. Her skill and competencies include laboratory cultivation, analytical assay and phytoplankton cultivation of phytoplankton in a photobioreactor system, microscopy, culture isolation, so very wide skills until DNA-based activities, electrophoresis, and so on. And she was at the algal training course on Fraunhofer in Stuttgart, called Algal Biotechnologies, Techniques and Opportunities for the Sustainable Bioeconomy. And she participated at the domestic symposium of PhD students in Zagreb, and so she is very active in the activity that belong to the PhD student in Zagreb. So again, thanks to the two speakers for their participation. And now I give the floor to Professor Narcisa Bandarra. Welcome, and please. Thank you.
Hi. Okay, sorry. Now I escape. Okay. Let's see if to start, but I realize, well, first of all, I want to thank you for this opportunity and for your kind words that Professor Concheta and also Maria tell about our presentation, and we will try to share with you the work that we are developing here in IPMA, and with some giving an overview of different topics that we have work. I don't know, at the moment I'm very small here, and I'm trying to be a little bit, but okay, the presentation is, but I don't, let's see. Well, I will try to follow you in the different slides, and of course these are a topic is bioprospecting and nutritional approaches, and of course marine bioactive compounds and health benefits. At the same time, as all we know, oceans have been showing rich habitats and great biodiversity and chemical entities that proven bioactivity and are so important in the disease prevention and treatment.
Narcisa, just sorry to interrupt you, Narcisa. You're not sharing your slides yet, just so you know.
No?
No.
Oh, what is something? But we have done it. Sorry, everybody, because we tried this before and it works very well, and now it's always the same. Okay, sorry. I hope it will work now.
That's great.
Yeah?
Yes.
Okay. And now I'm here also. Okay. Well, but again, I will go to the second slide. I think now it's okay. Okay, thank you. As was I telling you, the importance of the oceans and the importance of these biomolecules that give some bioactivities, and the so many applications that we have, and we try to get here in this slide the importance of all these compounds. Here we try to stress the main aspects that have to be taken into account in this area of bioprospecting. and at the same time, based, of course, in sustainability of new marine resources, because it's always in that we are focusing, and at the same time also look to underexploited marine resources that need to be upgraded. And of course, we need to have very good analytical tools that help us to identify and to detect new compounds with important applications, and in different areas, like it can be also in food, in the pharmaceutics, in biomedical, and so on, different areas. And we also have some different methodologies that work in vitro models that well, mimetize what happened in the digestion process, to have an account what happened when we ingest the different compounds and bioactives, and to look the different bioactivities. Here we follow the different areas related with marine sources, with the ingredients, and also these applications in food. Of course, this amazing work gives deep insights into the importance of marine-derived biomolecules as a future source for drug discovery, but not only, also in marine ingredients, and focus in food and feed, and this is areas that needs to be undertaken and explored in terms of get more information on that. Here I will share with you some data that we obtained in the projects that we are running, and in the publications that also papers that we published in these aspects. Look, for instance, for this microalgae, Skeletonema, that we have here the data concerning the ethanolic extracts, and look so high the level of anti-inflammatory activity. It means that it can have here important biomolecules that can help in the prevention of diseases, and this is something that we study in some projects. And for instance, Skeletonema was one of the microalgae that presented compared with others, and in particular ethanolic extract, a very, very high anti-inflammatory activity. And is only to show that among the different microalgae, you can have different responses. Everything depends on the type of extracts, on the type of solvents that we are used. All the time we try to use green solvents to be used also in food systems, and this is something that we use to do. Another example that we obtain here about Isophysis galbana, that is another microalgae that we study deeply. And what we realize here that, in fact, in a dry weight, there's very high lipid content, 27%, that is a very important level. And at the same time, the important omega-3 fatty acids that are always searching. And you can see here that 33% we have in these omega-3 fatty acids. And for instance, DHA, that we know the importance, and later on I will show you more data concerning these fatty acids. Presents 2.1 grams in these 100 grams of dry weight. That is a very, very important level. As you can see here, 21 grams of fresh microalgae in a perfect world, it will be okay to give you 500 milligrams of EPA in DHA. But it was if this microalgae presents 100% of bioaccessibility of each fatty acid. But the world is not perfect, and in this slide we can see that in fact the bioaccessibility of this fatty acid is lower than 50%. What means that you need to give new processing techniques, for instance, or increase the doses, because it's so far to this And you can see here in these results the low bioaccessibility. This is also some questions to share with you and to think about that we need to evaluate in this microalgae also. We evaluate the microalgae anti-inflammatory effect, for instance, again in the Isochrysis galbana, and we have seen that before digestion was a very high level, but it decreases after digestion. Is another topic that we need to think about, because sometimes all the process needs to be evaluated and all the line during when you ingest, but what happened, what is the bioaccessibility, what happens after digestion needs to be evaluated. And sometimes you don't have only this supplement is not enough. You need to follow the line and to understand what happened in your health. And this is another way to pay your attention to these aspects. Again, because also we work in some seaweeds. Here you can see seaweeds are not so important about lipid content, but they have very interesting extracts. And you can see here, for instance, for Cystosera, that it can present a very high level of antioxidant activity, in this case in aqueous extracts. Well, I will also want to share with you, as I told you before, DHA is a very important fatty acid, as all of you know, but is a very important component of our brain and represents around 40% of total fatty acids of our brain. It means something. It means that it is a very important component of cellular membranes. And also something that is important. With aging, it decreases, and we need to be attentive to this process and to this decrease. Well, usually we get this fatty acid from fish, but we need to show you, as I told you before, sustainable sources. And we try to look to some microalgae, and for instance, or Antioquitrum, that is a source that can give us some DHA with a very, very high level. Look, this 61% in dry weight, that is also very good. And we need to evaluate all the source and trying to see what happened. And this is, in fact, a very sustainable way to have DHA. And now I want to share with you a paper I have all sent at the here. You can see this was a paper that was recently published, but in Food and Function. And what we want to see there is the development of novel food products, of course, using this Antioquitrum, but not only the development of this product. I have to tell you that we use in this lean yogurt 2% of biomass of our Antioquitrum. But the results don't stop here. We want to evaluate nutritional value of this new food and also determine the digestibility and bioaccessibility. That are questions that all the time we need to evaluate. And of course, the bioaccessibility decreases, but is something to take into account, particularly when you want to determine the doses that are, for instance, for the prevention of cardiovascular diseases. And this is something that I want to share with you, because you can see the production aspects that in the biotechnology, a very important tool in this way, and also the development of all the steps, and also connecting with the bioaccessibility and connecting to this evaluation. I'll leave to you the reference of the paper, and if you need something more, we can talk about. Another area that I want to stress, and as we are talking here about the European Society of Biotechnology, marine biotechnology, I think that is important to stress these tools that are so important. For instance, as I told you, DHA, when it's located in this SN2 position of DHA, is more effective, is more bioaccessible. And at the same time, in this project, we prepare some new supplements based on that. And of course, because we want to evaluate what happens if we use some tools to prepare these structural lipids. These structural lipids were with the short chain fatty acids, saturated fatty acid position N3 and 1, and DHA and 2 and the SN2 position of tag to be more effective. And I leave you the paper that we present and the data that is here to help in the effect, but I will share also with you some data that we obtained that. And what we can see, particularly in the brain, I will pay attention you to the brain, that we increase the level, and this is something very, very difficult because its compounds need to pass the hematoencephalic barrier, and we know that all the times is very difficult, this effectivity. And okay, these are the data, and also we have good results related with the liver and also with the erythrocyte increase, that it shows that these structures can be very used and can try to be developed using biotechnology, connecting with all the different aspects. Here we have another paper, but more connected with adipose tissue. We know that the brown adipose tissue is the health fat tissue that we have in our body. And we want to evaluate what happened with this type of compounds. And as we can see here, this at green color and the right side, you can see that DHA in this brown adipose tissue, it increases, and this is something that also interesting, but it doesn't happen in a white adipose tissue. And we know that the brown adipose tissue, if it has a higher amount of long chain omega-3 fatty acids, is more healthy than saturated ones. For instance, after that, we can say that these new structures can be alternative dietary supplements for human diet and take advantage also of the higher DHA bioavailability and structural lipid forms. Well, to finish, I want to thank you all, the group that is working in this paper, but not only my colleagues in HIPMA, because these projects were international ones, and I want also to thank to Spanish Ministry of Science, because this project was coordinated for Almeria University in terms of structural lipids production, and this is something to take into account. And at the same time, I want to finish with a sentence that I like very much: the mind that opens to a new idea never comes back to its original size. And this is something that we are doing here. And of course, this sentence is from Einstein that means something. And at the same time, thank you very much for your attention. And of course, I'm here for answer to your questions. Thank you.
Thank you very much, Narcisa. It's a fantastic talk. I'm gonna, as I say, move on to our next speaker, and then we will take questions and answers at the end. Thank you very much, Narcisa. If you could stop sharing now, so then I think if you go, it should be, yes, perfect. So welcome, Ena, and if you would like to share your presentation, and we're more than happy to see what you have to present for us today.
Can you all see the presentation?
Yes, but can you please put it at full screen?
Yeah. Okay, I will. I just wanted
Perfect.
to. So before I start, I would just like to thank you, Maria and Concetta, for inviting me and for this beautiful introduction that Concetta gave. So I'm really, really grateful to be a part of this webinar. So thank you. So I will talk about biomass productivity and lipid accumulation of 3 marine microalgae that were grown in oil refinery wastewater. So basically, we all know that microalgae are photosynthetic organisms, and they're comprised of a diverse group of eukaryotes and prokaryotes with a really simple cellular structure. But what is special for them is that they have a great morphological diversity, that they're widespread, and that their size varies from 5 to 50 micrometers. Also, I would like to point out that they have a huge ecological importance because they're responsible for over 50% of global carbon fixation. But also what I will focus on is that they're important commercially. They're important in nutrition and in chemical and also in pharmaceutical industries. So they're valuable because of their production of valuable products or bioactive compounds. And those products are lipids or carbohydrates and proteins. On this table here, you can see a more detailed list of valuable products that microalgae produce. But I will focus on membrane lipids. So basically, we have membrane lipids that are comprised of polyunsaturated fatty acids, and they can be used as supplements for food and feed for animal nutrition. And storage lipids that are comprised mainly from C14 to C18 fatty acid, and they can be used as renewable energy source. Basically, since microalgae are so widespread and that they have ability and they have a high ability and tolerance to a wide range of environments, they can be used in they can be cultivated in wastewater as well, because wastewaters are rich in nutrients that microalgae use for their proliferation, but also they can biotransform pollutants such as heavy metals. And by that process, we get a process called phycoremediation. It is a biological treatment method of purifying the wastewater. And with that in mind, with that treatment method, what you get, what is a plus side for you, is that you have an increased biomass production and lipid content that you can use. So basically on this picture, you can see, there is like wastewater example from an oil refinery that you use, and from Adriatic Sea in our case, we isolated microalgae species that were put later and that were cultivated in photobioreactors with wastewater. And after a certain amount of time with certain conditions, you get an algal biomass that can be used in our case for biogas or biodiesel or bioethanol production. So the aim of this study was to determine the biomass and lipid productivity of marine microalgae species with an aim to estimate their potential for biodiesel production. So we have 3 microalgae species, Nanochloropsis gaditana and Picochlorum wilhelmi, that are picoeukaryotes, and Synechococcus sp, that is a cyanobacteria. So those 3 marine microalgae were isolated from the northern Adriatic from Rovinj, and they were cultivated in 50% diluted oil refinery wastewater. The batch experiments were conducted at 3 different temperatures, at 18, 24, and 30 Celsius, with a high light intensity of 130 micromoles, with light-dark cycle 12 hours, with 19 salinity. So basically, we determined the biomass with pre-weight polycarbonate filters, and lipid extractions were done according to Bandarra. Gas liquid chromatography was done for fatty acid analysis, and we had nutrient determination by spectrophotometry. So basically, I will just mention that the algae were cultivated in our photo bioreactor system. So basically, this is how it looks in our lab. So we have 4 photo bioreactors, and they have a LED panel or and luminometer around them. So it's kind of like a cape that surrounds the photobioreactor with light intensity that we desire, that we want. And we have a PhD and OD conductivity probes that are controlled by our with our SCADA system that is installed on our computer. And we have a CO2 and air blower. And we have a chiller and a cooler at the end that helps to maintain the temperature in the photo bioreactors. Our experiments are mainly conducted in those photobioreactors. So there is 4 of them. So when it comes to our results, we have here our picoeucaryotes Nanochloropsis gaditana and Picochlorum wilhelmi. When it comes to biomass yield, you can see that both strains, both species have the best biomass yield on 30 Celsius. And their biomass yield is pretty similar. It's 1.1 something grams per liter for Nanochloropsis gaditana. And for Picochlorum, we have around almost 0.2 on 30 Celsius. When it comes to 18 and 24, you can see significantly lower biomass yield. But here on 24 Celsius with Picochlorum wilhelmi, we can see a little bit higher biomass yield. But on Synechococcus, strain, we can see that that the highest biomass yield was at 24 Celsius. And other temperatures weren't that great. As you can see on the 18 Celsius, it was almost a flat line. When it comes to lipid content, so I put again Nanochloropsis gaditana and Picochlorum wilhelmi together because they're picoeucaryotes. So on 18 Celsius, we see the variation, but it's not that big of a variation. So it starts from 43% lipid content and it finishes at 26 on the last day of the experiment. On the 24 Celsius, we have also not big of a variation, and it kind of finishes at the same lipid content. But on 30 Celsius, we see the highest lipid content was at 60% at the middle, somewhere around the middle of the experiment. On Picochlorum wilhelmi, when it comes to 30 Celsius, we have a pretty significant growth from 15% to almost 48% of lipid content. Lipids at 24 were also pretty high. So we have from 60% to 30% almost. And at 18 Celsius, we have also really high lipid content from 56, but it goes down to 30% of lipid content on the last day of the experiment. But Synechococcus, we only conducted it at its temperature at 24. You can see clearly the lipid content starts from 32% at the beginning of the experiment, and it finishes on the 15th day at 15% at 24 Celsius. Our fatty acid profiling showed that Nanochloropsis gaditana has a significant percentage of saturated fatty acid. So you can see that their percentages here at 18% and on 24 and as well as 30 Celsius, it is pretty high. So it's almost to 40% and it's almost the majority of saturated fatty acid. But also you can see the monounsaturated fatty acid are right behind them with the lowest polyunsaturated fatty acid content. And when it comes to picochlorine wilhelmi, we see that we have as well a significant saturated fatty acid content. It varies like from 25 to 12% throughout the experiment. But basically what we noticed that there is a higher percentages of polyunsaturated fatty acid in picochlorine wilhelmi, which you can see by these charts that they vary like from 16%, 19%, and almost somewhere around 10% at 24 Celsius. And on Synechococcus, you can see clearly that saturated fatty acids are in the highest concentrations. So they are from there to 21%. And you have polyunsaturated fatty acid, which you probably cannot see because it's just a thin line here. There wasn't any polyunsaturated fatty acid in this experiment at this temperature. And when it comes to nutrient uptake, we measured ammonium removal at all 3 species at 24 Celsius. And you can see almost pretty same speed, let's say. Nanochloropsis gaditana took 5 to 10 days, while picochlorine wilhelmi took 6 to 13 days for a complete nutrient uptake. Synechococcus was a little bit longer. It took from 7th day to 15th day for a complete ammonia removal. So I will just conclude with this, that higher temperature and high light have a positive effect on biomass yield that can be later used for biogas, biodiesel, or bioethanol production. And analyzed picoeukaryotes have substantial biomass yield, while Synechococcus has less biomass quality and less biomass yield. And our Nanochloropsis gaditana has the highest productivity of lipids and saturated fatty acids that can be used for biodiesel production. That's why Nanochloropsis gaditana is a good model for biomass production, but also for a process of phycoremediation of industrial wastewater rich in ammonium and nitrates. While picochlorine wilhelmi has a significant percentage of polyunsaturated fatty acid when you compare it with Nanochloropsis gaditana, which can be used for a nutrition or an animal feed, but that is still something that needs further investigation and further experiments that need to be conducted to determine that. And with that, I just want to thank you. And yeah, I think I'm a little bit faster, but yeah.
Thank you very much, Ena. That's been a fantastic talk. Thank you for showing us your research and your so far results. So with that, I would like to ask the co-hosts if they can be open for any questions and answers from our participants. And I don't see any questions in the chat. As far as I can see. I'm pretty sure you have questions, but you just didn't potentially have the time to write it. I could potentially start with a question from Narcisa, if you could answer this. So you, with Skeletonema, the species that you extracted, you used the water and ethanolic extract. So you saw that the ethanolic extract gave you the highest yield. And do you know do you have any idea of what kind of compound you would find there? More like, obviously, yeah. This is a very, very complex answer, but do you have any impression, any group of compounds maybe?
Yes, at the same time, as you can see in the slide, I'm looking, we also evaluate the aqueous extracts. and the aqueous extract was not so good than this. And we realized that some lipidic compounds probably can be there because ethanol is able to extract some lipid compounds. And we believe that is not the best for ethanol, for lipids, but we believe that some lipidic compounds may be here and can have some effect. At moment, this was a project with a company, and at moment this extract is in evaluating related with the pain. They want to develop something to apply, for instance, in biomedical way to as a control, as a pain control. And they were at moment, I think, working on that. We have this work. Of course, we think that some lipid compounds can have this and can be there, and we also try to evaluate this extract that didn't come in. But of course, it's very difficult, and you know that to arrive to the biodiscovery and to identify the compound, it takes years and years and years. And at the moment, we start with that and we evaluate, and we know that EPA was there, some EPA was there, and probably, but not only, because we know that we have extracts, we have synergies. And sometimes we know that it is very complicated to evaluate. But thank you for the question. In fact, was something that we tried to get. We don't have not yet the compound that is responsible for that, but we hope in a future, not near, I will say, because it will take time, but the company at the moment are working also on that.
Perfect. Yes, I know it takes time, and especially the key is that you started and you find an interesting organism to begin with. So thank you very much. I think there's a question for Ena from the participants. Torger says, what happens to heavy metals in the wastewater? Will they accumulate in the final product, do you think?
By my understanding, they don't accumulate in the final product. I think they're just microalgae take an uptake of those heavy metals, and they form a biotransformation of them so that they're less toxic. I mean, I think I think that's how it goes. Basically, they use the process of biotransformation, and with that, you don't have heavy metals in your final products.
Would it worth to have a way of identifying this at the end product?
I'm sorry, what?
Would it worth in the future to look for any confirmation of any bioaccumulation of toxins at the end product, maybe as a next step or at another project, just to identify whether there is?
Yes, I would like to know. I would also like to know. Yes. Yeah.
Perfect. Any other questions? I have a couple of questions for Ena. Very, very interesting talk. Thank you so much again. So you, for the culturing, you use several certain conditions, and you varied mainly the temperature. Did you do any tests to identify, for example, the ideal light conditions, for example, or light dark? Or I know that sometimes people do have, they do grow algae, microalgae at continuous light, for instance. Have you tested that at all?
Okay, so basically, this research is a previous project that is done now. And basically what it was done, there was a lot of microalgae strains that were tested at different light conditions. So it was like 12-12, 16-8, 24-0, and other conditions like that, but it's still not done. And it's still a part of my PhD to see exactly that, to see the light duration, how does it affect. So basically what I'm doing is we're starting an experiment with complete darkness and with complete light all the time. So on all of our photobioreactors. So I'm really anxious to see what will happen. Yes.
Fantastic. I have a question from Roseanne. Hi, Roseanne. Nice to see you here today. S. marinoid, which is a species you mentioned, I think, can be tricky to grow. Do you think it could be a problem for drug development? I think it's. I think this is for Narcisa. I'm not sure.
Yeah, probably. Yeah.
Yes, Narcisa. Yes.
This Aurantiochytrium is not a problem to grow because it grows very fast. You need to have bioreactors. You need to have well, control conditions in your bioreactors. But compared with green algae, it grows more fast. I have to tell that 3-4 days, and you have a very high production. Aurantiochytrium is better than Taustochytrium. And probably the question is related with that because marinoid sometimes it can change. We know that these are heterotrophic microalgae, not the autotrophic one. And as we're heterotrophic, we give them a source of carbon source. And at the same time, as glucose, molasses, or something, subproducts of industry, also we had some work on that. And it can work well in the production, but of course, are not outside like for other autotrophic species that sometimes you can do this. It can you need to grow these species in bioreactors and everything very controlled in controlled conditions. But the time of production is it's okay. We know that some countries at the moment, this is a very alternative source for DHA. And we know that pharmaceutical companies and also some supplement companies are based on these heterotrophic macroalgae.
Great, thank you very much, Narcisa. I have another question from Concetta, actually. She asks, which model systems did you use to assess the anti-inflammatory properties, like cells or animals, and the bioavailability?
Sorry, I didn't get.
Which model systems did you use to assess the anti-inflammatory properties, like cells or animals? What kind of
cells
models? Yeah, and to assess the bioavailability as well. Yes.
Yes. To assess the. Thank you for the question. To assess the anti-inflammatory effect, we use in vitro kits and trying to evaluate the COX activity. It's based on that. But to evaluate the bioaccessibility, and we evaluate in vitro, also mimetizing, simulating what happened in digestion in mouse, and after what happened also in the stomach and in the intestine, trying to get all the line of your model, in vitro model. But sometimes it's not enough because you need to go to the bioavailability. And to have the bioavailability, usually we need to use in vivo models. Of course, this is getting, each time is more and more difficult to have in vivo models. We know the ethics and all the aspects related with the use of animals and so on, but we leave that for the last chance. But at the moment, it's not possible to evaluate some bioavailability without that, and also some aspects concerning, for instance, we are developing some projects trying to evaluate the effects in the Alzheimer's and so on related with HA. Of course, at that time, we need to have not only the cells, but also the animals at the end of the study to get some data concerning in vivo studies. I don't know if I answered, Conchita, to your thank you for, it's good to see you.
Yes, the same for me. Thank you so much. It is okay with your answer. Thank you very much for your
Thank
contribution.
you. It's
Thank
a
you.
pleasure not to be with you because now we are
Yes.
away, but hope to meet you soon
Yes, the same for us. Thank you.
in a face-to-face way.
Yes, in presence. Thank you.
As everybody understands, we're all suffering from lack of face-to-face meetings, but I think these webinars are a good chance to bring everyone together and discuss about our research and developments, and make sure that research is going on, it still continues, and communications are still being out there. So I think there's another question from Ena, from Concetta. We still have a couple more minutes to go on with some questions. So Concetta is asking, did you find correlations between biosynthetic pathways of fatty acids and antioxidants? And that would be either negative or positive correlations.
I don't think we I don't think so. I'm sorry, I don't think so. We still haven't determined that. I'm sorry.
Of course, no. I think there are lots of things in the PhD that you can think and incorporate in the future, or next steps in research.
Yes.
That's why I think Concetta is very right to ask these things, because then it triggers a few thoughts that we can move on to, to enhance a bit more our understanding. Concetta, do you have any other question for Ena on that?
No, it is okay. This was just only an explorative question. As usually we know that the two biosynthetic patterns can show some peculiarity or positive or negative correlation. So it was only intended to explore this aspect, but of course there are many aspects related to grow biomass and lipid biosynthesis. So the presentation of Ena was very full of details. Thank you very much.
No problem, anytime.
I have one last question for you, Ena. I saw that you have the growth curve of your model organisms, and some of them continued longer days, some of them stopped earlier. Is there a reason for that?
Honestly,
if I remember correctly, I might be wrong.
Yeah, you're right. I'm still not sure of that reason. It was just basically we were waiting for it to get to the exponential phase of growth, and people were like, okay, it's done now. So basically, it was just mainly the process of our thinking was like, okay, it's done, so people were still learning, and this is just the beginning. So my growth curves that I'm doing now are much longer, much longer. Yes.
Okay, yeah, I mean, in a PhD, in the beginning of a project, everything optimizes with time.
Yes.
And you have fantastic results already, and thank you so much for sharing those with us. And of course, good luck with the remaining of your PhD, and thank you both, Narcisa and Ena, for sharing your work with Anna as well, who is your PhD student and was part of this work as well. And I think if we don't have any questions, I would like to close the session, this ESMB webinar for today. And please, I will be advertising more in the future. I am Dr. Maria Salta, and I'm based at the University of Portsmouth. I'm a board member and a secretary of the ESMB. And if anyone wants to reach to us to perhaps, you know, have some work they would like to share with us, please definitely do so. And this series is started now, I think, as a need for communicating in the pandemic, in the time of pandemic. But there is absolutely no reason for this not to go ahead after things open up, because we find that is extremely useful. If you have any questions, or you would like to communicate with any of us, in our website, there's all this information, and this recording will be uploaded soon. And if you want to contact with any of the researchers today, or you have any further questions, please go ahead and reach out. If anyone else has no questions, I will close the seminar, and thank you all very much for being part of this today. Thank you and have a lovely day.
Okay, thank
Bye.
you. Bye, thank you.
Thank you. Thank you very much.
Thank you.
Thank you.
Okay.
Bye. Bye.
Bye.
Thank you. Thank you.